Okay, this is today we're going to tackle chapter 10 and chapter 11. Chapter 10 is about vision, and chapter 11 is about muscles. So here we go. I think if I can get this thing going. There we go. Okay, vision, chapter 10. The whole area that you can see without moving your head or eyes is known as your visual field. And some people have a wide visual field, and some people don't have a wide visual field. Mine's pretty big. It all depends on how far your eyes stick out of your eye sockets. So if they stick out pretty far, then you've got a pretty good visual field. It's about 180 degrees. I have a brother who has tunnel vision in his visual field. His eyes are set back in his head, so his visual field isn't nearly what mine is. Um, while the visual field may be wide, an individual's visual acuity tends to be only in the middle of the field of vision. This is because there's a concentration of cones in the middle of the visual field, an area known as the fovea. So the cones are the receptors in your eyes that detect color. Uh, there's rods and cones. The rods detect light, or just weak light, relatively weak light, but it's mostly white, and it's mostly white light that it detects, and then cones. I was downstairs in my basement. I was running on my treadmill, and in the morning, and I turn off the lights when I leave the basement. And I saw a yellow light. I saw a yellow light when I turned off the light, and I couldn't figure out where the yellow light was coming from. It turned out that I was seeing out of the corners of my eyes, I was seeing a light on the bottom of the treadmill that was actually red, but I saw it as kind of a whitish, yellowish light because, of course, I was seeing it with my rods, not with my cones, and I couldn't detect the color, as interesting and not funny as that is. Okay, so this is what it looks like if you look at a picture with your fovea. And this is what it looks like if you look at it from the side. You really can't see it very well, and it's fairly fuzzy. All you're seeing is light and dark. However, even with the concentration of visual receptors in the center of the visual field, there is a spot where the optic nerve feeds out of the eye and blood vessels feed into the eye. This is known as the optic disc, and this area maintains a blind spot in an individual's visual field. Normally, the brain automatically fills in the blind spot. If you've ever if a doctor has ever examined you, one of the things they will do is they will look at the blood vessels in your eye. One of the things they're trying to figure out is whether it's normal or not. You can detect diabetes by looking at the blood vessels. Diabetes tends to make your blood vessels brittle, and people actually go blind when they are diabetic. Some people will go blind. Usually, the treatment is so intense that you can maintain yourself for an extended length of time, even with diabetes. As you can see, the blind spot is not in the middle of the field. That's the fovea right there. That it's off to the side in both your right eye and your left eye. So what a doctor is doing, he's trying to look at the health of your blood vessels in your eye. He's not just trying to detect diabetes, he's also trying to detect glaucoma, anything that would burst a blood vessel in your eye. That's not really supposed to happen. These blood vessels are supposed to be fairly healthy. Vision comes to humans in the form of wavelengths of light or photons. The human visual ability lies between the 400 and 700 nanometer, ultraviolet to infrared. And as you can see, 400 is black and 700 is fairly black as well. That's ultraviolet, ultraviolet meaning it's so violet that it's black, and infrared is also black. Other animals have ranges of vision that encompass wider spectrums of light. Bees can detect beyond ultraviolet light, and that's one of the reasons why they can detect where another bee has flown because they can see the trail of that bee, and that's what they will follow that trail back to a food source. A photon of light comes into your eye with select properties that we will perceive as colors. The hue and the wavelength of the light, wavelengths in the 400 to 500 range, are very dark purples to blue, and that's right here. Wavelengths in the 500 to 600 range are greens to orange, with yellow in the middle, and that is right here. Right here. Wavelengths in the 600 to 700 range are bright reds to darkening to black. Whoops, I'm sorry. Let me go back to where we were. Yeah, there we go. Reds to black. 700 is right there. Since most color is detected as patterns of light of different wavelengths, brightness refers to the amount of white light from the object. Saturation has to do with the amount of light of a select wavelength. Of all the photons reflecting from an object, if they were all 510 nanometers, the object would be highly saturated as green, and this is 510 nanometers actually, and as you can see, it's a very intense color of green. The eye acts much like a camera. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye, that changes the light beam as it enters the eye. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscles. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. And of course, if you're moving a piece of paper away from your eyes or towards your eyes, of course, you would need to accommodate that piece of paper so that you could read the writing on that piece of paper, and that's known as accommodation. And of course, it's the ciliary muscles that are changing the shape of the lens. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction, which means that in the first grade, about 15% of the males and 18% of the females will be wearing glasses. In some populations, it's higher than that. Some groups, genetic groups, will have more people that have eye problems, and so they'll wear more glasses. And in other groups, you will have fewer. By age 20 until presbyopia is when you get old eyes. That's what presbyopia means. 48% of females and 32% of males require correction. That means 1/3 of all the people, of all the males in the United States who are 20 years and older, 20 years and older until they get to be about 45 or 50 years old, about 1/3 of them need glasses. It's about half of all females between 20 and 45 to 50 will need glasses, as interesting as that is. My daughter wears glasses. My son doesn't. He does now, but he's almost 50 years old. So he wears reading glasses, but he has never worn glasses. My daughter started wearing glasses in her 20s. Nearsightedness or myopia is where the eyeball is too long and the nearest focus occurs before the light hits the retina, and that's nearsightedness. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. And astigmatism is a misshapen cornea. It's elongated, that causes the light to pass incorrectly into the eye, and it causes the individual to be nearsighted. So people that have astigmatism are nearsighted. The iris is a muscular covering under the cornea that gives the eye its color. Non-brown eyes were a mutation that appeared about between 6,000 and 10,000 years ago. So 10,000 years ago, if you saw a hominid, if you saw a human form, I think we were Cro-Magnon men at that time. Everybody would have brown eyes. And then there was a mutated form. Blue eyes was a mutated form. Those people became popular, I guess, and so they reproduced, and now we have a relatively large percentage of the population that is blue-eyed. And I'm not sure what the percentage is. I'm going to look it up right now. The pupil is an opening in the iris that allows light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles. Let's see if I can get this thing to work. The photoreceptor cells of most animals, let me see. Wait a minute. Let me look this up. Well, you know what? Why don't you guys just watch me do this? Why not? Instead of go on to, there we go. Whoops. All right. All right. Yeah. Well, let's go on to another one. Here we go. Okay. Percentage of the world's population that don't have brown eyes. Well, all right. 79%. Between 55 and 79% of people worldwide have brown eyes. Brown is the most common eye color, which means that between 45 and 20% have blue eyes, or green eyes, or gray eyes, or hazel eyes, or whatever. But most of the world's population have brown eyes. Almost all Asians have brown eyes. That includes the Chinese and the Japanese and people from India. Very few non-brown-eyed people. Mostly the blue-eyed people are Europeans. Africans don't have blue eyes, for example. But the Europeans seem to be the only ones with non-brown eyes. So if you have brown eyes, then congratulations. Both my kids have brown eyes. Then again, my first wife had brown eyes. Didn't she? Yeah, she had brown eyes. Photoreceptor cells of most animals are wait a minute, did I do this correctly? Is that right? Yeah, okay. The photoreceptor cells of most animals are part of the retina on the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. The first layer is the optic nerve fiber layer. This is working from the inside out. The second layer are the ganglion cells. The third layer are the bipolar cells. The fourth layer is the actual photoreceptor cells themselves, the rods and cones. And the very last layer on your eyes are pigmented epithelium. And the reason you have pigmented epithelium is because it's highly reflective. And since it reflects, it tells the rods and the cones how much light there is. And that's, you make up this huge picture from all these little pieces of light. Now you can see the pigmented epithelium at night if you shine an eye into a light into somebody's eyes. And the reflection you get back is the pigmented epithelium. And as you can see, you've got this herd of cats right here. There's a tiger and a Siamese cat. That's a cat as well. But here's a deer right here. And the interesting thing is that you can almost tell what species you're looking at from the color of the reflection of their eyes. And as you can see, here are all these cats, and they all have different hues of pigmented epithelium. Here's one right here with its eyes closed. Anyway, the rods and cones detect light that reflects off the pigmented epithelium. As I said before, the rods and the cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve, and then that impulse is sent back to the brain. While most of the communication in the retina runs from outer layer to the inner layer in a vertical pattern, amacrine and horizontal cells communicate with cells in a horizontal pattern. The horizontal cells connect receptor cells. Amacrine cells connect the bipolar and ganglion cells. And if something happens, like you get a flash of light, the amacrine in the horizontal cells, instead of destroying your uh, receptor cells, they will short circuit the whole vision thing, and they will keep you from going blind. The rods and the cones represent different functional systems. The scotopic system consists of all the rods in each eye. There's about 100 million cells, rods in each eye. So you have 100 million rods. That's 200 million in both eyes. That's a lot of rods if you think about it. And this is the scotopic system. The scotopic system is very sensitive to weak light but cannot detect color. As I said before, I was seeing that light from the corner of my eye, and it looked to me like it was yellow, maybe it was white. When I looked for it, I couldn't find it. It turned out that it was a light that when you turned off my treadmill, it went from red to green. And of course, I couldn't detect the red or the green. All I could detect was the yellow. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside the fovea. The photopic system consists of all the cones in the eye, about 4 million. So you've got 100 million rods, and you've got 400 million cones in each eye, which makes 208 million pictures that you're getting. And you take all those little pieces of information and you make them into a picture. And that's what's happening to you every nanosecond. You're creating a picture in your own mind from all these little pieces of light, all these pixels of light. The system detects color and requires relatively strong stimulation, and therefore is primarily a day vision system. The photopic system is located primarily in the fovea, but is present in other areas of the retina, but in a lower concentration. There are 3 forms of photopigments called opsins that detect the 3 color groupings. The photopigment for the scotopic system, of course, is rhodopsin, and we've already said that. The photopigment for the photopic system is one of the 3 configurations of opsins. All the photopigments in the eye are made up of 2 parts, retinol and opsin. Retinol is short for vitamin A aldehyde or retinaldehyde, shortened to retinol. Retinol and opsin. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site on the opsin molecule. The enzymatic opsin molecule combines rapidly with G protein transducin. Transducin in turn acts on phosphodiesterase or PDE to convert cyclic guanosine monophosphate, cyclic GMP, to 5-GMP. Cyclic GMP inhibits sodium channel closing, and with the conversion, the sodium channels close. The loss of sodium ions depolarizes the neurons, just like in any other receptor. Since we're dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as to the amount of light that you are detecting. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are 2 types of bipolar cells that transmit information: off-center bipolar cells and on-center bipolar cells. On-center bipolar cells are activated when light is turned on. Off-center bipolar cells are activated when light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when light is weak or turned off. Glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. Each cone in the fovea is connected to 4 types of bipolar cells. one on-center midget bipolar cell, one off-center midget bipolar cell, several on-center diffuse bipolar cells, and several off-center diffuse bipolar cells. Each midget bipolar cell connects to just one cone in the fovea. Each diffuse bipolar cell connects to several cones in the fovea. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. The retina will create a mosaic of stimulation that the brain puts together as a picture. Now, the interesting thing is that with some of your cells will react to weak light and some will react to strong light. When you close your eyes, if you're looking at a picture and you close your eyes, you get an after image. That after image is from the weak light. That's the off-center image that you're getting. And it's usually the opposite because you've fatigued your eyes. You have to look at a picture or you have to look at something for an extended length of time. Then when you close your eyes, if you're looking at something red, all of a sudden you see green. And that's because you have made all of the cells that see red, you've fatigued them, so you've used all that rhodopsin up. That opsin up, actually, retinol. You've used all that retinol up, and as soon as you close your eyes, then the opsin becomes the dominant, and the only thing that's left in your eye, and so that's why you see an after image. And it's always the opposite color on the color spectrum of what you've been seeing. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds. This is known as range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination. Calcium is a major player in photoreceptor light adaptation. When an individual changes from one extreme light to another, it takes time to adapt to the new intensity of light. This is because the retinol and opsin break down at a higher rate in bright light and must regenerate to be available in the dark. So the reality is that if you go out in the dark, because you've been in a situation where you can see light, when you go into a dark situation, your eyes will eventually adapt, and it doesn't take that long. All you have to do is wait for about 5 minutes, and your eye you can see relatively well, not all that well because there's not a whole lot of light coming in. But after 15 minutes, you can see very well, relatively well. There's still no light coming in, so you just have to wait to adapt. Strangely, most humans don't want to do that, so they don't like to go out at night. Of course, you guys have a taboo against it, but if you go out at night and you sit there for a while in the dark, pretty soon you can see fairly well. Adaptation is much quicker when adjusting from dark to light because the retinol opsin level is not depleted, so it's much easier to go from dark to light than it is to go from light to dark. You can be fairly blinded when you first go outside, and of course that's kind of scary, but if you just wait for a couple minutes, your eyesight will get better and better and better. This is one of the reasons why, if you've ever watched one of those movies where the guys are wearing the nighttime goggles and somebody showed a light in their eyes, of course, they were blinded and they couldn't see for a couple 10 or 15 seconds, and it's because it used up all the retinol and opsin, and now suddenly, of course, they are blinder than they would be if they hadn't been wearing their night vision goggles. We're not exactly sure, and they're doing research. Our military uses night vision goggles. When I was in the military, they didn't have night vision goggles. Well, they did, but it wasn't for the lowly likes of me. Of course, it was for spatial ops and whatnot. So, but now a lot of people wear night vision goggles, and it seems to be causing problems. So we'll get more information on that as um, we get more people coming back from Afghanistan and Iraq who had to wear the night vision goggles in order to raid places at night. Color acuity is more accurate in the center of the visual field because of the fovea is almost exclusively cones. Moving out from the fovea, the number of cones decreases and the number of rods increases. Light sensitivity is most acute about 20 degrees off center, where the concentration of rods is greatest. And of course, in the military, when I was in the military, they used to teach you at night, if you were in the dark and you're trying to see something, that you not to look directly at it, that you needed to look at, I don't know, it's about 20 degrees off center. And then you'll probably detect it. This is one of the reasons why people see ghosts. A lot of times what they're seeing is they're seeing light out of the corner of their eye. When they focus on where the light is, of course, now they're focusing the cones, which don't really detect light that well. They detect color, and they were using the rods before, and of course that's why they were able to see that weak light. And then when they tried to look directly at it, it disappeared. And of course, if they looked away from it again, then the light came back. So that what they were detecting, this concept of a ghost. About half of the primary visual cortex, the V1, is devoted to the fovea. This is one of the reasons that humans are able to concentrate their spatial field on tasks close at hand, such as reading and surgery. A scotoma is a gap in a perceptual field, usually due to injury of the visual mapping area of the brain. People with scotoma will often compensate for their problems by allowing the part of the brain not affected to dominate the affected area. This is known as blind sight. They just fill it in. They automatically fill it in. And that's what we see here. This is them when they first look at the picture. They've got a blind spot. And after they've been looking at the picture for a while, it actually fills itself in. And we can do this ourselves if we concentrate on this red X right here, and we continue to concentrate on that. This gray spot right here will slowly go away, and it will fill in with this pattern of this pattern. And it looks like that pattern is moving. But the longer that you stare at the red X, the smaller that gray area will become. It will fill in with that pattern. We're not going to do that. Each eye is divided into two parts: the nerves of the temporal half of the visual field, nerves of the nasal half of the visual field. As the optic nerve leaves each eye, the nerves come together in an area known as the optic chiasm. It's right here. Here's your eyes. The optic nerve comes together, and this is the optic chiasm right here. As the optic chiasm, the nasal optic nerves cross to the opposite hemisphere, and the temporal optic nerves stay on the same side. After the optic chiasm, the nerve pathways are known as the optic tract. Most axons of the optic tract terminate in the lateral geniculate nucleus of the thalamus. From the lateral geniculate nucleus, the axons radiate into the primary visual cortex, V1 of the occipital lobe. And as you can see, this is the pathway that it takes. The primary visual cortex is sometimes called the striate cortex because of striations that run through the area. Visual areas of the brain not in the striate cortex are known as the extrastriate cortex. Some axons of the optic tract send their axons to the superior colliculus, where the superior colliculus helps the eyes to track rapid movements. And of course, my grandson was playing football today, and he made an interception, which was kind of interesting. But the reason he was able to make that interception was because of his superior colliculus, which allowed him to track the ball through the air. He was behind the guy. They passed the ball to him, and it was low. So he went down for the ball. He actually dove on the ground and caught the ball. He's only 8 years old. It was quite a sophisticated catch. The lateral geniculate nucleus is arranged in 6 layers. The outer 4 are the parvocellular P layers, and the inner 2 are the magnocellular M layers. And as you can see, here are your 4 P layers, and here are your 2 M layers. They are darker. The P cells have small receptive fields, while the M cells have a large receptive field. M cells are sensitive to low contrast and rapid change. About 10% of the retinal ganglion cells are M cells. 80% of the retinal ganglion cells are P cells. P cells require high contrast to be stimulated. P cells are relatively insensitive to rapid temporal change, such as flicker. P cells are sensitive to different wavelengths of light, and they therefore detect color. P cells have sustained responses. P cells have high acuity. And this is one of the reasons why when you're looking at your computer screen, you don't see the screen flickering. It is your brain fills that part in, even though it's flickering. But if we took a film of your computer screen, we would see the screen flickering. The primary visual cortex is the first and most abundant area in the human brain. From the V1, the stimulus is farmed out to other areas of the brain, depending on what parts of the eye the stimulus comes from. Areas are designated from V1 to V5, and you can see them all right. Well, you can't see all of them. You can see V1. Not exactly sure why it looks like that. V2, V3, and that's V3 as well. Researchers have discovered that there are 3 different kinds of cones. Cones that are sensitive to short wavelengths only, cones that are sensitive to long wavelengths only, cones that are sensitive to middle wavelengths. And as we talked about before, you have cones that detect blue, cones that detect green and yellow to orange, and ones that detect red to black. Of the middle wavelength cones, some are more sensitive to the higher ranges of middle. Some middle range cones are more sensitive to the lower range of middle. There are twice as many long wavelength cones, the reds, as middle wavelength cones, the greens and yellows. Short wavelength cones are the fewest, that's the blue and purple. So strangely enough, when you ask people what colors they like, everybody's favorite color tends to be red. Seems to be the dominant favorite color, and then green, and then blue, as odd as that may seem. Myopia is a condition where the eyeball is elongated, causing the image to focus on in front of the retina, causing a blurred image, and this is what the image looks like. There is some evidence that nearsightedness is caused when children look at images too close to their faces. When I was a kid, we used to get real close to the television set, and my mother would tell us to back off, to back away from the television set. Now I don't I don't get anywhere close to the television set, but my mother taught me well, and of course my vision was fairly good. It still is pretty good. When I was in the military, I had 20/10 vision, which is pretty good, which is the best, I guess. They wanted to make me a helicopter pilot. I guess I was smart enough not to do that. Anyway, myopia, this can be prevented by only allowing reading at adequate light, avoiding small type, reading without glasses anytime that you can do so. And of course, this is what myopia looks like. Everything's fuzzy, and as you can see, they're getting fuzzier and fuzzier and fuzzier. Now they're going to put on corrective glasses, and maybe not. Oh, there it goes. Okay, now they're okay. Glaucoma is a group of eye conditions that damage the optic nerve, the health of which is vital for good vision. This damage is often caused by an abnormally high pressure in your eye. Glaucoma is one of the leading causes of blindness for people over the age of 60. And as you can see, as glaucoma, the pressure increases. The blind spot, actually, this is scotoma. This is a scotoma forming. And eventually the fovea is the only area that you can see out of. And there you go. There you go. And that should be it. Yes. Okay. Now we'll start on chapter 13, motor control and plasticity. Motor control and plasticity. Okay. Muscles cover the skeletal system and provide movement. They also provide protection for the organs that lie underneath. How muscles attach to the bones is an indication of the type of movement that they perform. Muscles only provide force when they contract, when they tighten up or get smaller. And this is one of the reasons why, in order to make your bicep strong, you've got to move your arm in, and of course it contracts the muscle and makes it look bigger. And it actually turns hard at that point. Some muscles maintain body posture. Some muscles produce movement around a joint. Any movement that occurs in the body is performed by muscle. Muscles are connected to bone by tendons. Around a joint, there are two sets of muscle groups. When one group contracts, the other extends. These muscles are agonists, and an example of an agonist would be your tricep, this group of muscles on the back of your arm, and your bicep, that mass of muscles on the inside of your upper arm. Tricep and bicep. Muscle groups that act together to perform the same act are known as synergists. Muscles are made of thousands of fibers. These muscle fibers are made up of two kinds of filaments. Thick and thin filaments arranged in a regular pattern. Thick filaments are made up of myosin, and the thin filaments are made up of actin. Of course, those of us who have looked at raw meat, meat is actually muscle fiber from animals, and we eat it. Why in the world would we ever do that? Why would we eat animal fibers? And the answer is because it's protein, and we need the protein. And we're going to talk about how you build your muscles in just a second. There are two basic types of muscles in the human body. Smooth muscles contract in an automatic fashion without human control. These are smooth muscles. They're regulated by the autonomic nervous system. Your heart muscle and the muscles in your digestive tract are examples of smooth muscles. The other type of muscles are striated muscles, primarily supporting the skeletal system. This is the stuff that I had a chicken sandwich for lunch, for dinner. That's the only meal I've eaten, so I guess that's it. Anyway, the chicken was striated muscle. That's what it was. And you could actually see it if you pull it apart. You can see the muscle, the individual muscle fibers. You can see the same thing in steak, except, well, we won't go into that. Since different muscles perform different tasks, each requires different speeds, precision, strength, and endurance. Striated muscle fiber come in two forms, fast muscle fiber and slow muscle fibers. Really kind of weird. We've been talking about this for, golly, how long? About 40 years, 50 years. No, we were talking about this when I was running track in college, and that was over 50 years ago. Fast twitch muscles and slow twitch muscles. People that can run long distances and have a lot of stamina, they have slow twitch muscle fibers. People that are sprinters, they have fast twitch muscle fibers. Some people have a combination of both. They tend to be your better runners. But then again, you'll come across a guy who seems to have no muscle mass at all, and the guy's got all the stamina in the world, and he can run relatively fast. He runs marathons. He doesn't really have a sprint to him, but he can run really hard for long distances. Eye movement must be quick to follow the movement of speeding objects, but only for short periods of time. Extraocular muscles are fast muscle fibers. Fast muscle fibers react promptly and strongly. They fatigue rapidly. They are used when muscle tension must change frequently. Slow muscle fibers have greater resistance to fatigue. They are used mainly to maintain your posture. Muscles react to stimulus coming in from motor neurons. The axon will split into many branches and activate many muscle fibers. As the axon is activated, it releases the neurotransmitter acetylcholine. Acetylcholine makes the muscle react by opening sodium and calcium channels in the muscle fibers. The ionic change allows the muscles to contract. The point where the muscle fiber and nerve structure communicates is the neuromuscular junction. The neuromuscular junctions are large and very effective. And there's a neuromuscular junction. Each neuromuscular junction releases enough acetylcholine to not only cause one reaction, but several at the same time. One cause of muscle fatigue is due to diminished effectiveness of the neuromuscular junction. Muscles develop coordination when axon terminals migrate to specific target muscles and acetylcholine receptors migrate to the clustered axon tips, allowing for more efficient muscle reaction. And we're going to see a video of this right in just a minute. Muscles react because spinal or cranial nerves send messages to the muscles through efferent neurons. Motor cells of the spinal cord are larger and faster than other motor neurons. Generally, small motor neurons activate slow muscles and are more easily excited. Large motor neurons tend to activate large muscles, which require more stimulation for activation. And now we're going to watch that video. There we go. There we go.
The skeletal muscle of animals, including humans, are striated. They exist in bundles of thousands of muscle fibers. A single muscle fiber is actually a muscle cell. As you can see, muscle cells are very elongated and contain several nuclei. Packed inside the muscle cells are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. When a motor neuron stimulates a muscle fiber, overlapping filaments in the sarcomere slide along one another, and the sarcomeres shorten. The repeated pattern of the sarcomere results from the arrangement of the two major proteins within the myofibrils: myosin and actin. The myosin heads are the actual mechanism for muscle contraction. Now let's examine the process in detail. First of all, when a motor neuron stimulates a muscle fiber, calcium ions are released from the sarcoplasmic reticulum. The ions force the tropomyosin to displace and expose myosin binding sites on the actin. An ATP molecule is unhydrolyzed, and the myosin head forms a cross bridge. When the ADP is replaced by a new molecule of ATP, the myosin head detaches from actin and goes back to its original state. And that's pretty much the whole process. And one more thing. Each myosin head attaches to and releases from actin independently, not in unison. Otherwise, the myosin heads would all release at once, and the whole muscle would go to a relaxed state. And that wouldn't be good.
Okay, and that's how muscles work. The body must maintain control over the position of its parts. This knowledge of the position and state of its muscles and limbs is called proprioception. This knowledge is maintained by select receptors that report muscle and joint positioning. Muscle spindles lie parallel to muscle fibers. Golgi tendon organs report on the position of the joints and are situated with one end attached to the tendon and one end attached to the muscle. Muscle spindles are located in many areas of our skeletal muscular fiber. Muscle spindles are actually spindle-shaped. They consist of an afferent element and an efferent element. Their primary function is to maintain their posture and tell you when you have been in a position too long. And that's what they look like. You can see their spindle shape. When a muscle fiber is activated, the muscle spindle reports rate of change. Rate of change refers to the change that the fiber has gone through to be able to function. The muscle spindle also reports force, the force required to maintain contraction. And of course, anytime you lift something, what did I lift that was heavy? I was cutting we had the derecho that blew through about 2 months ago. And we're still cleaning up. Actually, I just finished cleaning up, but I chopped down the last tree stump that we needed to chop up, and I had to pick up the stump, and it was about 2 inches in diameter, and it was about 4 feet long, and it was a piece of maple, and of course all woods are different concentrations. Some of them are heavy, and some of them are not that heavy. We have a willow log or willow tree that most of the logs were a little bit lighter than the maple. Maple is a much more concentrated wood. So when I picked it up, of course, I was carrying it to the wood pile. And I had to determine whether I could actually carry it, the 50 feet from where I cut it to the wood pile. Or would I have to put it down? And that's the force that we're talking about. That has to do with the rate of change that we're talking about. Muscle spindles have 2 types of receptor endings. Primary sensory endings are wrapped around the middle of the fiber. They're activated during the initial phase of contraction and then adapt to a lower discharge rate. Secondary sensory endings are found near the smaller ends of the fiber and are activated at first, but only after prolonged use. And here are your secondary endings right here and here, and this is your primary sensory endings in the middle of the muscle. Muscle spindles are informed of impending action through 2 special motor neurons. The gamma motor neurons, or the gamma efferents, inform of change of position from the initial movement. I've got a cat that's running around all my pills. Gamma motor neurons, or gamma efferents, inform of change of position from the initial movement. It comprises about 30% of the efferent fibers. Alpha motor neurons perform initial movement. 70% of the efferent fibers are alpha motor neurons. While individual muscle movement is monitored by the muscle spindle, muscle contraction is monitored by the Golgi tendon organ. Golgi tendon organs are able to monitor muscle joint movement because joint movement is controlled by tendons connected to muscles. And that's what a Golgi tendon organ looks like. So if you lift a weight, the weight's a little bit too heavy for you, the Golgi tendon organ will tell you that you may damage your muscle. You may pull your muscle away from the bone, and of course, it tells you not to do it. Golgi tendon organs are somewhat insensitive to passive muscle stretch because they're connected to the elastic component of the tendon, and it is only activated under heavy loads. The Golgi tendon organs detect overloads that threaten to tear muscles and tendons. Activating the Golgi tendon organs inhibits the motor neurons supplying the muscles that pull on the tendon, and thus, by relaxing the tension, prevents mechanical damage. And of course, that is what it's supposed to do. One of the problems with using steroids is that sometimes if you use steroids, and you work out a lot, what will happen is that your muscles will get too big for your bones. Your muscles are only supposed to be a certain size, and there is an optimum size and a minimum. Well, there's no minimum size, of course, but there's an optimum size. And so, just because you take steroids doesn't mean that you can get these huge, bulky muscles. Sometimes the muscles will be so strong it will pull the muscle away from the bone. It will break the tendon. It has to be reattached. My daughter was had a roommate who was a weightlifter, and this was actually in Tempe. Yeah, she lived in Tempe, and so so he claimed that he was all natural and that he wasn't using any steroids. And then one day he was at the gym and he pulled a pectoral muscle, which is really hard to pull. I mean, they're almost impossible to pull because they're attached to your rib cage, and so they're not really they're attached very very well and in several different places. And he actually tore his pec. And at that point, of course, she knew that he was using steroids because you just don't do that. I mean, you don't try to lift a weight that is so heavy that it tears your pectoral muscle away from your body. It just doesn't happen. Your muscle's not that strong. So if you're trying to bench press 500 pounds, for example, normally people don't bench press 500 pounds. But if you're on steroids and Arnold Schwarzenegger, when he was Mr. Universe, could do that. But uh, a normal person can't really bench press that much, and if they try, they'll damage themselves, and that's exactly what happened to this guy. He tore his pec, and she knew he was on steroids, and she yelled at him for it, and he apologized. The whole 9 yards, the whole thing. Anyway, okay, this is how we build muscles.
Muscle building. The key to building muscle is protein synthesis and cellular repair. Skeletal muscle is made up of long fibrous chains containing proteins. Bundles of thousands of fibers make up the muscle itself. When an athlete engages in a heavy workout, this structure is damaged, causing micro tears in the muscle fibers. The body naturally repairs this damage by mending the torn fibers. The result is a muscle that's larger and stronger than before. And with each additional workout, the athlete adds bulk, strength, and speed.
Okay, we're going to watch this one as well. Wait a minute. Let me stop this just for a second. Okay, so we saw that the way you build muscle is you tear your muscle and then you repair it. It gets a little bit bigger, and then you do it over and over and over again, and now all of a sudden you've got big muscles. Okay, so why in the world do steroids make people why does it make people bulk up so much? The answer is that steroids allow the body to repair the damage that is done very rapidly. So normally you should allow your body to repair before you start lifting again. So I can lift today and I can lift in 2 days because my body will have repaired itself. My muscles aren't sore anymore. I can go back to the gym and I can do the same thing over again and build my muscles bigger and bigger and bigger. So I need 2 days of rest to repair and to build muscle. If I don't do that, my body will break down. However, if you take steroids, what steroids do is they allow your body to repair themselves very, very rapidly. So instead of lifting every third day, you can lift every day. You're taking steroids, and when you sleep at night, as long as you're taking in a high-protein diet, then you can go out and lift again tomorrow. You can lift the next day. And because you're able to increase your workouts, you're tripling your workouts. Because you do that, your muscles get much larger. Now, of course, as I said before, if you use steroids, there's always a possibility that you will create a muscle that is too strong for your body. It is bigger than it's supposed to be. So what will happen potentially is that you'll tear muscle away from bone. And if you do that, it's a pretty good indication that the person is on steroids. So this is how we know that baseball players, if they tear a muscle, if they tear a muscle away from a bone, we know that they've been on steroids because that muscle is bigger than it's supposed to be. Normally, you can't do that. You just can't do that. Your bone, your tendon, your Golgi tendon organ will tell you that you're trying to lift too much, and it will set the limit at that point. And this is the reason why, if you look at bodybuilders all have pretty much the same bone structure. They have the same muscle structure. Really tall and slender people cannot do that. They can't really build muscle. The best that they can do is well, they can't get really bulky like The Rock. You know, The Rock is a big guy. He's a really big guy, and he's got really big bones. So he can put lots of muscle on top of those bones, and that's why he looks like he does. Arnold Schwarzenegger, when he was Mr. Universe, was the same way. He was a big guy, so he could put lots of muscles on that big frame. And now that he's not lifting anymore, if you look at him, he's kind of flabby. And the reason he's kind of flabby is because he's got those big bones. And of course, even if he doesn't eat very much food, even if he's not taking in the protein that he was before, he can't fill it. He can't fill all that skin up with muscle anymore. Anyway, okay, so let's go ahead and see what this TED Talk says.
Muscles. We have over 600 of them. They make up between 1/3 and 1/2 of our body weight. And along with connective tissue, they bind us together, hold us up, and help us move. And whether or not bodybuilding is your hobby, muscles need your constant attention because the way you treat them on a daily basis determines whether they will wither or grow. Say you're standing in front of a door, ready to pull it open. Your brain and muscles are perfectly poised to help you achieve this goal. First, your brain sends a signal to motor neurons inside your arm. When they receive this message, they fire, causing muscles to contract and relax, which pull on the bones in your arm and generate the needed movement. The bigger the challenge becomes, the bigger the brain's signal grows, and the more motor units it rallies to help you achieve your task. But what if the door is made of solid iron? At this point, your arm muscles alone won't be able to generate enough tension to pull it open, so your brain appeals to other muscles for help. You plant your feet, tighten your belly, and tense your back, generating enough force to yank it open. Your nervous system has just leveraged the resources you already have, other muscles, to meet the demand. While all this is happening, your muscle fibers undergo another kind of cellular change. As you expose them to stress, they experience microscopic damage, which in this context is a good thing. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle-building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger.
Okay, it's talking about cytokines here, and cytokines, of course, are what's released to tell your body that it needs to repair something. Strangely enough, cytokines are what's released when somebody has COVID-19. They're released and they tell the body that it needs to prepare itself for an inflammatory process. If you have too many cytokines, then the inflammatory process becomes extreme and the person goes into crisis. And that's one of the things that they're complaining about with COVID-19 is that you're producing too many cytokines. This is one of the reasons why, I don't know if you were listening to when President Trump was hospitalized, but one of the things they gave him were steroids. Now, steroids reduces your immune response, and the reason it does that is because it tells you that you need to build muscle. It tells you that you need to repair things really fast, and because of that, because of that repairing process, it lowers your immune response. So one of the reasons that they're giving people steroids who are in a severe situation, like they can't breathe, is because the cytokines are causing a overabundance of this immune response. So they give you the steroids to slow down the immune response. Steroids, of course, do a lot of different things. One of them, they raise your norepinephrine level, and you get these ideas of grandeur, which is really kind of curious. And it can cause, well, it causes roid rage. And I think, and of course, that's what we think is maybe going on with President Trump. He's having this reaction to the medications that we gave him to keep him from suffering from the potential damage to his lungs by the coronavirus. Really fascinating stuff. They're talking about cytokines as far as building muscle is concerned. You produce cytokines anytime that you need to repair damage. A couple days ago, I was using an axe, and I stretched the ligaments in my hand, which is a sprain. I've sprained both of my hands by chopping with an axe. And now, of course, it creates cytokines. My hands are swollen to some extent, and now they're being repaired. But eventually, of course, in about a week, my hands will be fine. But it's the inflammatory process that builds muscle, and it also helps repair your body.
As they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, those generally don't produce enough stress makes muscles bigger and stronger. As they damage to the muscle tissue that activate the immune system. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle-building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger as they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, those generally don't produce enough stress to stimulate new muscle growth. So to build new muscle, a process called hypertrophy, our cells need to be exposed to higher workloads than they are used to. In fact, if you don't continuously expose your muscles to some resistance, they will shrink, a process known as muscular atrophy. In contrast, exposing the muscle to a high degree of tension, especially while the muscle is lengthening, also called an eccentric contraction, generates effective conditions for new growth. However, muscles rely on more than just activity to grow. Without proper nutrition, hormones, and rest, your body would never be able to repair damaged muscle fibers. Protein in our diet preserves muscle mass by providing the building blocks for new tissue in the form of amino acids. Adequate protein intake, along with naturally occurring hormones like insulin-like growth factor and testosterone, help shift the body into a state where tissue is repaired and grown. This vital repair process mainly occurs when we're resting, especially at night while sleeping. Gender and age affect this repair mechanism, which is why young men with more testosterone have a leg up in the muscle building game. Genetic factors also play a role in one's ability to grow muscle. Some people have more robust immune reactions to muscle damage and are better able to repair and replace damaged muscle fibers, increasing their muscle building potential. The body responds to the demands you place on it. If you tear your muscles up, eat right, rest, and repeat, you'll create the conditions to make your muscles as big and strong as possible. It is with muscles as it is with life. Meaningful growth requires challenge and stress.
That was actually what this one is, the same thing. So there you go. That's how it works. Let's see if I can make this thing work. There are two reflexes that are maintained by the spinal cord that are automatic and are not mediated by the brain. The flexion reflex is activated when pain is applied to the leg. The muscles will contract, moving the affected part away from the stimulus. And this is a video showing the reflexes of the body. Oh, no, it's not. This is...
The Hulk is an anthropomorphic exoskeleton which mimics the human form. It provides extra support that enables a person to carry more weight than they normally could. Exoskeleton technologies are considered part of the robotics field. If you want to think about them as wearable robots, that's pretty accurate. They sense what the user wants to do, where the user wants to go, and mimics the motion. We like to call these autonomous systems. The exoskeleton is primarily composed of titanium components, which are both lightweight and provide the strength needed to carry the load. A microprocessor takes in readings from the sensors that are throughout the structure and calculates where the user wants to move, and then commands the actual hydraulic system to actuate the joints to provide the motion. You can walk, you can run, you can bend, you can crawl, you can leap. It does not impede your range of motion whatsoever. This technology could be a real benefit to the military. Just imagine you're a soldier operating at 6,000 feet in the Afghan mountains and being asked to take 120 pounds up in that level of thin air. How exhausted you would be once you got there. An exoskeleton provides the ability to carry that weight the same distance, but to have energy left to execute the mission once you're there. We've also looked at attachments that allow users to carry weight not only on their back, but also on the front. The exoskeleton will fit anybody between 5'4" and 6'2". The current system weighs 53 pounds, but it's pretty much unnoticeable to the user because what the technology does is it even transfers its own weight to the ground, in addition to the weight you're carrying. And we're taking the load off the soldier's back. And we're going to help prevent injuries that occur from carrying heavy loads and repetitive lifting tasks. The best part about working on this technology is that it gives us a glimpse into the art of the possible.
Okay, I can't remember why I showed you that. I've got another video coming up that shows you the reflexes. Interesting, if you look at most military veterans, I guess, if you look at most veterans, the injuries they have are usually to their lower back or to their knees, and of course this exoskeleton could potentially take care of all of that. The knee jerk response, the quadricep muscle that is contracted to allow the knee to bend is informed of the tendon pressure that makes the muscle react to, by stretching, thus the stretch reflex. And we're going to see a film of that. Where is that? There it is. Okay, I don't know why it's here. Okay, let me finish this. The primary lateral cortex of the M1 controls all the muscle movements in the body. The hands and the lips utilize the biggest portion of the area compared to the percent of the body that they represent. And as you can see, there are different areas. So if you put a probe and you touch these different areas, you will the muscles will react to that. Most of them are hands and face, or lips actually. Your lips are very sensitive. The M1 communicates with the rest of the body through the pyramidal system. The pyramidal system is a triangular-shaped bundle of nerves that runs from the brain through the medulla and into the spinal cord. Lesions to this system will result in the inability to move select joints and limbs. Those neurons that run from the M1 to the muscles that are not in the pyramidal system are said to be in the extrapyramidal system. These neurons control the spinal reflexes, and now I have a video about the spinal reflexes.
The lower body reflexes will be performed in the same manner as the muscle strength exams were performed. We're going to test one side and then quickly test the other side and compare side-to-side responses. We've got 5 reflexes that we're going to go over, one being the knee reflex, one the Achilles reflex, and then the adductor reflex, the internal and external hamstrings. The first one will be the knee reflex. I'm going to have the patient close their eyes, strike the tendon. We got a good response. I want you to keep your eyes closed, strike it again. Got another good response. Now the Achilles reflex. I'm going to just slightly dorsiflex the foot by reaching underneath the foot and letting her rest the foot on my hand. And then I'm going to reach back and I'm going to tap the Achilles tendon. And I got a real nice plantar flexion of the foot. Comparing side to side, I have the patient lay face down. Now we can do the adductor, internal and external hamstring all in this position. If I'm going to test the adductor, I'm going to have the patient medially rotate the leg. I find the adductor tendon along the medial side of the leg. I've located it, and I'm going to strike over the tendon, and I get a slight contraction of the adductor. For the internal and external hamstring, I just have the patient lay flat. I locate the tendon by slightly having the patient bend their knee against resistance, bring the leg up. I'm going to push down. And the tendons pop out quite easily. I've got them, you relax the leg. I've got the tendon located, the medial hamstring tendon. I'm going to strike the tendon, and I'm watching for contraction and slight flexion of the knee. On the lateral hamstring, I locate the tendon, and I'm going to do the same thing, strike the tendon, and watch for contraction of the hamstring as well as slight flexion of the knee.
Oh, there we go. Okay, we don't need to watch that. That's all right. Okay. Oh, they were talking about being in the mountains of Afghanistan at 6,000 feet. Those of you who are living in Lukachukai or Salih, you're at about 7,200 feet. I'm sorry, 6,000 feet. So you're about 1,200 feet higher than what he was talking about, the thin air of Afghanistan. So you guys are actually 1,200 feet higher than what they're talking about. 1,200 to 1,500 feet higher. The extrapyramidal system communicates with the spinal cord through 2 tracts, the reticulospinal tract and the rubrospinal tract. The extrapyramidal system not only handles spinal reflexes, but muscles responsible for breathing and the muscles that control the head and the neck. And that is it. Okay, so I will type up the quizzes for these 2 chapters. Next week we will tackle, we will tackle chapter 12. So next week is just chapter 12. And chapter 12 is a fun one. It is about, sex. It's about sex, evolutionary, hormonal, and neural bases of sex, and that it's always interesting. So there you go. Don't tell Professor Barber that I'm stealing his topic. He's the one that teaches human sexuality. as weird as that is. Okay, so I'll see you guys. Stay safe, and I will. Don't worry about your midterm grades. If you have done anything, then I will give you a passing grade. The only people that will not pass are the people that haven't done anything at all. So there you go. And I will get those in. You have paper that is due in the beginning of November. This is the second Saturday, second Sunday of the month. So you've got about 3 weeks, 3 or 4 weeks to get your papers in. So there you go. And I'll see you next week.